Good morning once again. This is the second part of microbial genetics. I was trying to explain um, the process of gene transcription, and I told you that there are basic four types of four stages of gene transcription. The first stage being initiation step, followed by elongation, followed by termination, as well as post-transcriptional modification. So, ever before in the first part, I have been able to actually explain that this is the double helix structure of DNA, and being the double helix structure of DNA, the DNA will be acted upon by RNA polymerase to be able to undergo the process called transcription. And I told you that the there are four basic types of polymerase, RNA polymerase. In case it is in prokaryotic organism, it is RNA polymerase 1 without any other further subdivision. But if you are dealing with eukaryotic organism or eukaryotic cell, we talk of there are three basic types: RNA polymerase 1, which is going to produce ribosomal RNA; polymerase 2, which is going to produce messenger RNA; as well as pol 3, which is going to give us transfer RNA. So I said that transcription proceeds from 5 prime to 3 prime end, and that is the reason why we have this red part of the strand that is actually represented at this level. At initiation step, I made mention of a Tata box. That is the region at which the raw material for this process of transcription we actually proceed from. So it is this starter box that the transcriptional factor is going to bind at promoter region of starter box. So, and I told you that if we are dealing with prokaryotic cell, this starter box is made up of 10 sequences, and if it is eukaryotic organism, it is around 70 sequences. So, and that process continues. The next thing after the binding is to unwind the DNA to be able to expose the region of the messenger RNA for further binding. And the next one is going to be that elongation stage, at which complementary strand of this RNA will be synthesized, where the U will be replaced, where T will be replaced with uracil in that. And at elongation, just like I said, at elongation, the RNA will continue to move towards 5 prime end to 3 prime end. So, and if it is moving in that direction, you will see that the what we have here, we have 5 prime, which is the upper one, is called the template, DNA template. Okay, we'll make use of this DNA template when we go to the laboratory technique. By the time we'll be talking about PCR. So after that DNA template, the second strand is going to be the complementary DNA, which is going to complementary strand, which is going to be from 5 prime end to 3 prime end. So without wasting our time, let's proceed straight to the third stage of transcription. Third stage of transcription, and that is termination stage. So it is at termination stage that the RNA polymerase will move on towards what is called a terminator sequence. And the terminator sequence is basically made of. So there will be this. Okay, I said termination. The RNA termination stage. So the that I said we move from 5 prime end towards 3 prime end we now migrate towards this terminator region, and that terminator region is going to be rich in GC, is going to be rich in guanine and cytosine. And not only that, terminator can be, there are two types of conditions that will terminate transcription. So terminator can be intrinsic, that is, if it is spontaneous, it can be intrinsic. Then it can be as a result when the RNA polymerase encounters a row segment. It is called row segment or row factor. So termination can be initiated at that point in time. So you should not forget that once it has, the terminator region means that it is going to be rich in stop codon. It's going to be rich in stop codon. Don't forget that I dedicated one video for how can we learn the triplet codon. How can we learn genetic code? How can we learn them? So I want you to revisit that video so that we're able to know that you should be able to know what are the stop codons and what are the start codon as well. So, but you should not forget that termination is when will take place when the RNA polymerase has actually encountered the terminator region, and this termination can either be intrinsic or it can be extrinsic. That is, it can be dependent on what is actually known as by encountering row factors. Okay, so now the next stage, immediately after it has encountered stop codon, that means is going to release matured messenger RNA will be released after termination, after it has encountered either row factor or if it is spontaneous. So what is going to happen is just that the messenger RNA will now be released into the system, and this is going to be part of what is going to be the difference between prokaryotic termination or transcription and eukaryotic transcription, so or gene expression. We are going to see this distinction as we go on. So immediately after transcription now, you know, transcription has ended. So the messenger RNA now, that is now the product, will now undergo the process of translation. And the process of translation is called protein synthesis. So protein synthesis, we will not be able to go into the details of the process of translation, but you should know that that is the stage at which the amino acid will be synthesized from messenger RNA. At this point, you should not forget that I said, depending on the RNA polymerase, there are three different types of three different types of RNA. We have transfer RNA, messenger RNA, as well as ribosomal RNA. Each of these perform different functions. They perform different functions during the process of protein synthesis. And you should know at this point in time that this is very, very important. Okay. Ribosomal, at protein synthesis, ribosomal RNA, they play a very vital role in protein synthesis. So let's begin to now look at the role played by each of these RNA, the role played by each of these RNA in translation or during the process of protein synthesis. The first one we are going to look at is the role played by transfer RNA. What are the roles that they play? Okay, transfer RNA, the structure of transfer RNA, the first thing is to be able to understand how does transfer RNA, how does it actually look like. So transfer RNA has a structure like this. Okay, so the structure at this point is the transfer RNA, and at this point of transfer RNA is the segment that is called the anticodon. Okay, it is that segment that is called the anticodon. So for example, if you have AUG, going back to the genetic code, you know that AUG is standing for methionine, for male methionine, okay, which is regarded as start codon. It is start codon. So on that, this is messenger RNA, mRNA, that is being synthesized from the previous transcription, the messenger RNA. So whenever you now have, don't forget that this occurs in triplet codon, so that AUG, so this is going to be U, this is going to be A, this is going to be C. So this place is called the anticodon, and on the messenger RNA you now have the codon. So it's just like a lock and key rule. Okay. Immediately after that, let's now look at what should be the structure of the function of transfer RNA. I told you that the first thing that the transfer RNA does is that it exposes the region for binding. So at this level, this is AUG. So at this other part, you have methionine, because this one is coding for methionine. So you have it. Immediately after this was completed, this one will be shifted, and that's called frame shift. We are going to see more of this as we actually go on, but you should know that transfer RNA, that is what it does, because it is the one that contains the anticodon that is going to bind at a messenger RNA. Okay, now the process of translation, that is a protein synthesis, can be divided into three, just like transcription, that you have it in form of four. So this one also has initiation stage. It has initiation stage, okay, elongation, which is the second stage, as well as a termination stage. So basically, we produce, this one will occur under, okay, there are base occur. That is the first process with the activation of amino acids. Okay, then the second one is transfer of the amino acid to where? To ribosomal RNA to ribosomal RNA, or to transfer RNA. Transfer RNA. So just like I told you that this structure of transfer RNA, and you can see that it is the transfer RNA that contains the anticodon, and it is this anticodon that will be binded with three-letter code on messenger RNA, and immediately this one has the process of synthesizing methionine. This one will leave the stage, followed by another one, followed by the next, followed by the next, and that is going to be the procedure at which it is going to do. So now let's now try and be specific and examine each of these according to what will take place. So at transition stage, at initiation stage, there are two basic activities that will take place. The first one will be that the messenger RNA that has been synthesized, the messenger RNA that has been synthesized from transcription will bind. It's going to bind ribosomes. It's going to bind to ribosome in the cytoplasm, and what that one, after the binding, that means there will be production or synthesis of the triplet codons that is going to, and each code